right. I'll do my best, and I think it's not that complicated. So I'll try to make it succinct and easy to understand for this audience. Thank you so much, Val, for that introduction. Thank you, Jennifer, for having invited me to this wonderful, very timely event. And I'm saying timely because it is the area of anti-aging and longevity. There's more and more academic researchers in the space of aging, and there's more and more interest from the industry as well, as you have probably heard in the media. I'm going to talk about this idea that the immune system really is taking the lead in driving aging of different organs and systems. And it was only in the year 2000, so very recently, that we started to think that the immune system has a role in aging. We know that it protects from infections. We know that if it goes awry, you can develop autoimmunity. But the link with cardiovascular disease or cancer or Alzheimer's disease or even depression is relatively new. We didn't know the biomarkers, we didn't know the mechanisms, but there was this idea that inflammation, for example, increases with age, right? And that may cause some of the most feared diseases of aging. And that's my research. And I'm going to tell you two stories. One that relates to how we study the immune system to understand mechanisms of aging and intervene. And the other one is related to an accelerated model for aging, and that is humans in space. So with that, I'd like to just quickly go over my disclosures. I have a lot to cover today, so this slide I'm going to just try to be very succinct and skip for the moment. So this first picture here that you see was published in 2013 by Lopez-Otin in Cell, saying these are the 9 hallmarks of aging. And I read through the entire paper and I tried to search for the word inflammation. It's not even mentioned once. So the idea that inflammation participates in diseases of aging, again, is relatively new. So I think that is largely missing here. And so just as a way of illustrating this, we conducted this in silico experiment where we are just searching for PubMed keywords between inflammation and aging, aging and senescence, other hallmarks of aging, cellular senescence, mitochondrial dysfunction, all that stuff. And then we score these papers and compare how many are published in each of the 2 types of keywords. And the immune system is actually the winner. So the first plot represents the number of papers that have been published in the intersection between inflammation and aging. And the other hallmarks of aging are listed as other plots right there. And then you see the p-values are all very significant, meaning that the immune system is actually now taking this lead and really determining rates of aging in different systems and organs. You probably all have heard that inflammation is important for aging. But again, this is relatively new. It's not the type of inflammation that you see when you cut your finger. That's the type of acute inflammation. It gets red, swollen, warm. Those cardinal signs of inflammation are one type. It's very different from what we're studying here. For the acute inflammation, there's canonical markers, very easy to measure in the clinic. For systemic chronic inflammation, this causes collateral damage in tissues and organs and increases the likelihood of developing all these diseases of aging that I'm going to mention. There's no canonical biomarkers. Please don't bring CRP. That has no value when it comes to diseases of aging. And so this is a new plot that came out 2023. It's the new hallmarks of aging by the same researchers, and they included inflammation. Now, there's more than 2,000 publications to believe that inflammation is not only one hallmark of aging, but it's actually related to all the hallmarks of aging and is triggering other hallmarks of aging. We published this paper in 2019 putting out the idea that inflammation participates in many diseases associated with older age, but also in adolescence and across the lifespan. And so we have demonstrated by multiple means that inflammation, chronic inflammation, is a trigger for cardiovascular disease, for depression. We published a paper in 2014 that we looked at depression disorder, major depression disorder. We also published in immunosenescence and other diseases that are listed here. So how do we study this thing? The first comment here I wanted to point out is that we don't study animal models. The reason is they don't translate easy. So we focus on humans. And how do we study humans? We try to measure as many parameters as possible, hundreds of thousands of parameters from blood, very accessible. Then we use machine learning and AI. That's the multi-omics part, right, Paul? And then we use integrated approaches to make sense of the data. And so this is what we put together with Mark Davis back in 2008. A strong clinical core that recruits individuals, a technology center, the Human Immune Monitoring Center. And then we have a strong bioinformatics core. With the right immunological expertise, we're able to develop new hypotheses and new therapeutics for age-related diseases. So the 1,000 Immunomes was created back then. We've been crunching the data and publishing a number of different papers. The 1,000 Immunomes consist of 1,000 individuals that went into this study. It's a longitudinal study of aging. We secured more than $50 million from NIH, plus 18 that were just recently funded, both for Stanford as well as the Buck Institute and Technion in Haifa, Israel. We measure everything under the sun except for hair length. And we started, one important finding was that the inflammasomes, which are these molecular scaffolds that enable the maturation of two important cytokines, IL-1 beta and IL-18, are increased in older adults. And it's really interesting because when you separate older adults already after the age of 60 into inflammasome high and low, they individuals differ by clinical status, by many other types of clinical endpoints. So hypertension, arterial stiffening, and even longevity. So these individuals with inflammasome high are more at risk of developing other types of conditions. And so we had now a lot of data, and we decided to create a compact representation of inflammation. And what am I referring to? Is we use a deep neural network, so AI type of approach, to derive what's called the inflammatory age metric. And I could have my inflammatory age be higher than my calendar age or lower. And once we do that and try to, I'm going to just skip this slide, it's a little bit too math heavy. But suffice to say that once we derive that scoring system for inflammaging, we can see that the age-adjusted IH, the inflammatory age, is predictive of multimorbidity. It's also predictive of frailty 7 years before it kicks off. This was predicted frailty score based on the inflammatory age metric in 2010. And this is the actual observed frailty score in individuals that are 75 years and older, measured in 2017. So we see a pretty good prediction of frailty. We can also, because we run a large number of different assays in these immune cells that are circulating in the body, we can correlate high levels of inflammatory age with low responses in multiple T cells and B cells that are circulating in these 1,000 individuals. And we also can correlate that with the frequency of CD8 T cells, which is a sign of immune aging. So high inflammatory age, low immune response to multiple stimuli. We had this collaboration with a group in the University of Bologna to gather data from centenarians. And we run our inflammatory age metric in centenarians, as compared to a control group, and you can see that they're on average 40 years below their calendar age. And so they're protected from disease for the most part, and they are also protected from inflammation. And so this was really too good to be keep it in the academia, and I'm going to mention how we translated that into a company. And so we can also predict ventricular remodeling, arterial stiffening, and then also we can predict mortality. So we built a gene expression surrogate of inflammatory age because we didn't have a lot of data in mortality in our study. And so we used this gene expression surrogate of inflammatory age to go outside of our wheelhouse and take public domain data sets. Looking at the Framingham heart study, we were able to predict mortality using this new metric of health, really. And so we got funded to create this startup with Professor Mark Davis and trying to look for interventions for inflammatory age. We run our first clinical trial, and this is a double-blinded placebo-controlled trial using this type of method that pools different compounds from the literature. We have 150,000 compounds, and then we do interaction analysis with gene expression and protein expression. So in other words, what we do is to try to identify a set of compounds that will normalize the level of your proteins, and so inflammatory age can go down to a healthy normal state. And so we actually pulled about 260 compounds that could target a particular immunotype. So let me explain that a little bit better. I could be 45 in my inflammatory age, and my neighbor could also be 45, but the way we get there is different because the way the algorithm works, or the way my exposure is causing insults in my inflammatory age level. I could have high levels of CXCL9, a protein, and my neighbor could have eotaxin being too high. So the interventions will be personalized. And so we run our first study in collaboration with an industry partner. And what I'm showing you here is the number of different compounds that are selected using our algorithm that are targeting CXCL9. CXCL9 drives cardiovascular aging, and we want to bring it down to decrease the levels of inflammatory age. The interesting part is that many of these compounds can be found in foods, and we can map that to foods and food servings. Sometimes it's a little bit difficult to do so because the applications are not very straightforward. Sometimes for certain compounds you need to take 400 servings of broccoli a day. So it's not, obviously it's not very easy to implement. But we put these compounds in pills and give them to our study participants. And these are the results of the first study that we did in the experimental group versus placebo. So what you can see here is after 2 weeks of taking this intervention, inflammatory age goes down significantly, as are the levels of protein eotaxin. And eotaxin has been associated, you probably know it from allergies and overreactive lung responses to asthma and things like that. But it also has a role in neurodegenerative disorders. And so if you bring it down, you're protected, more likely protected from developing dementia and Alzheimer's disease. And so we are able to bring it down significantly, and now we decided to go for a more immunotype specific that I mentioned before, and run this large study in 1,000 people. And this was a study that was designed to look at every single parameter that we know in terms of aging outcomes. And the data is still under analysis. But there was something really interesting because we took photos of every study participant here. And why we did that? Because we also want to address skin health. And what we did was to implement a photo age in these study participants. And you can see the predicted, the predicted age based on the image so on those photos, implemented in our PML My Age trial that I mentioned just now, and the chronological age. So we're doing pretty well at predicting age. When my grad student pulled this photo from the book, he said, "Oh, you look like 37." He got a bonus. And then that was a few years back. But the interesting part, really, really super cool, is that now we can correlate the level of inflammatory age with the skin quality. So I'm not showing you that in this slide. Actually, that's a different application in brain aging. But we have preliminary data to show that photo age can be used as a proxy for biological age. And I'm going to just leave it there. There's a lot of effort in our lab to predict inflammatory age, to predict immune endpoints just by looking at your selfie. So that's underway. So very quickly talk about brain aging. We build a clock for that. By the way, we build clocks. I want this to be very informal presentation, and I didn't mention, but we're in the business of building biological clocks as well, from transcriptomics, metabolomics, proteomics. We have all sorts of clock. And why do we do that? Because we want to understand whether an intervention can help in reducing aging, and so you don't have to wait 20 years to see the outcome. So you have surrogate endpoints. That's our clocks. But we also understand mechanisms. If you pull the genes or the proteins that are most important, you can actually gather and have some idea of the mechanisms of aging and hopefully intervene. So in this case, I'm showing you a clock that we built based on animals. So it's the first time we're using animals. But in this case, we're using mice, 15 different data sets, 400 samples, chronological age and transcriptomics age for the brain. This is a brain tissue that has been profiled, looking at many hundreds of thousands of genes. And here I'm showing you the correlation between age acceleration, this concept of age acceleration in the mice and in humans. So you can see, in this case, brain aging seemed to be very well represented in the animals. So very interesting findings are shown here, where you can see this model for Alzheimer's disease that carries a mutation, or a number of mutations that increase beta amyloid and tau tangles in the brain, are actually accelerated in their transcriptomics age. So the 5X-FAD model shows acceleration in the transcriptomics age. So now we're looking at the intersection between biological age of the brain and Alzheimer's disease. So is that correlated with what you see in humans? Yes, that's also correlated with what you see in humans. That's shown here. So all this is human data. And for the most part, the genes go in the same direction. So we now have a metric to measure the effect of interventions, but also to better understand the mechanisms of brain aging and dementia. And again, using a similar approach to pull compounds or drugs that can reverse these signatures, we were able to identify these 10 top candidates that are able to, again, reverse the signatures of brain aging. And we're testing these in animals and also in brain organoids, that's human organoids, to be able to, in maybe 18 to 24 months, submit this to the FDA as an IND. So I'm going to switch gears a little bit and talk to you about this model for accelerated aging that can be obtained from looking at humans in space, but also ground analogs of space. So about 2.5 years ago, Susie Sanello reached out to me. She used to be working in the Johnson Space Center in the Human Research Program in Houston for the agency, for NASA. And she told me, David, your research in the immunome and aging seemed to be very aligned with what we do in astronauts, because they seem to suffer from a number of different aging phenotypes. They have arterial stiffening, very rapidly after a space flight. They have cognitive issues. They have loss of muscle mass. They have all these things that seem to be associated with aging and largely due to inflammation. And so why don't we join forces? And I had a few meetings with her, and after maybe 4 or 5 meetings, I realized that she really wanted to pass the torch. She said that suffering from this terminal cancer, and she was moving more of an admin to an admin role. We have a grant together from NASA, we asked many questions that have to do with where humanity is going. I got to know her family, became very good friends, and she passed away in December of last year. So this is really a personal thing, mission for me. And one of the things that we were discussing over and over is this question I'm going to pose to this audience today, which is the same question that Gerald O'Neill posed in Princeton in 1969. And that was a question he posed to his students. He was a physicist. And the question is a planetary surface the right place for a rapidly expanding technological civilization? And I think the answer is obviously not. We have to conquer the space and reach the stars. And it is inevitable that we will become an interplanetary species. Elon Musk is saying we'll be in Mars in 2024. That's probably over-optimistic. But NASA is saying that in 2030 we will reach Mars. But this poses a huge challenge. And the reason is there is an acceleration of aging in space. And so we demonstrated that by different means. I'm going to show you some of the data. But we also created the first consortium for space health and human longevity to fund research in the intersection between these two rapidly expanding economies. So the government doesn't really help much, and that's shown here, right? 0.3% of the funds are dedicated to biology and physiology at NASA. NIH is only putting about 0.5% in aging research, and that's the very good justification to create a philanthropic centered consortium that has the mission to fund research to centers of excellence across the United States and also in Europe. In the UK and France for now. And the idea is to enhance human health both on Earth and in space. So we have this mission to positively impact humanity by enhancing human resilience for space exploration and longevity. We will be funding research in the intersection between space flight-related pathology and regenerative pathology. Everything that falls outside of this intersection is not going to be considered. And just as an example that really there is an acceleration of aging in space, this is a plot, or a table rather, that shows different risks associated with spaceflight and with aging, and the rates at which they decline or increase in this plot here, or rather table. And so on average, what you can see is that aging gets accelerated in space by 30-fold. So for each year you spend in an ISS, you will age around similar to what you would age 30 years on the ground. Very concerning. Now, when astronauts return to Earth, there is a rejuvenation process. So there are naturally occurring compounds that we're studying currently. The data is largely available to us via, luckily we have now the private sector taking over with SpaceX and Axiom and other companies that are in the area. And why am I saying luckily? Because NASA has been a little bit challenging to convince that, so I'm going to just give you an example. We order, or we request for samples of serum from astronauts that went into the, back to the Apollo mission. They're considered national treasure. So what are they good for if you don't study them? We can't access those. So now with Inspiration 4 and Axiom 2 and Polaris Dawn, all these missions that are going from private sector, we're having a huge amount of data that's available to us. So this is not just a scientific question, but also has economical implications. The phase 1 of this consortium will consist of the different organizations that are listed here. We have a first agreement with the Buck Institute, and phase 2 will also incorporate many other organizations, and we have some individuals listed there that we want to engage with, including, of course, Stanford. I'm showing you here the management team and the board of directors. Chris Mason is my co-founder of this consortium, as well as the company that we built. I should say we build companies as well. I'm an academic entrepreneur, but why it's not that I have a particular enjoyment or that I enjoy building startups, or the fever of Silicon Valley, reached to me. I think it's because the scheme, basic science to bedside, is completely broken. We can't get to that translational aspect of our research with federal money. There's so much we can do because it's relatively small, the grants are small, but also most importantly because they're reducing time. They're only five-year cycle, right? So we need to bring a company, a private investment into the equation. So basically, in my view, basic science to company to bedside. And that's the way we have been operating. And I'm going to show you some of the results we have now for the new company we formed, which is called Cosmica, which is effectively using space biology to resolve or solve chronic disease. And I showed you a similar slide before, but I would claim now that this is really the first non-disease model of human accelerated aging. Why am I saying non-disease? Because we've been studying progeria as a model for accelerated aging. That's a very different, those are very different processes than what you see in normal aging. HIV has been somewhat shown that accelerates immune aging. Some other pathologies like that, those are all diseases. In this case, we're looking at healthy humans that go in space, suffer from this process of accelerated aging, and then go back to Earth, and they somewhat recover for the most part. And so if we study this in a longitudinal fashion, having a large number of measurements, we can decipher mechanisms of aging at an accelerated pace, and we can also intervene and discover new drugs and also different nutraceuticals that can work for this thing. Can I know how am I doing with time, Jennifer? I'm good. Okay, wonderful. Okay, ahead of schedule. Okay, that's good. So I'm going to tell you about a few other things we have after I finish this discussion. So there are some methods to mimic what happens in space. So these are called ground analogs of space-related research. So, for example, radiation, which is not a really big deal if you go to the ISS, but yes, if you go above and beyond ISS, so say the Moon or Mars, you would be exposed to showers of particles that are really galactic rays, very detrimental for DNA health, right? So that can be simulated in the Brookhaven National Labs that are in New York. So we have now an agreement with the national labs there to irradiate organoids and to irradiate peripheral blood mononuclear cells to see the effect of galactic rays in these neural organoids. So those are mini brains. I'm going to show you some of that research. Cardiac organoids, immunological organoids that we get from tonsillectomies, and that they respond pretty good to the flu vaccine when you run an assay. They actually mimic what happens in vivo. And we expose this to galactic rays or simulated galactic radiation. And we also have a way to mimic what happens at the level of gravity. So there was this machine that was developed by NASA. It's called a rotating wall vessel that keeps the organoids or the cells in free fall. So effectively subtracting gravitational forces. And there you have a ground analog of microgravity. The European agencies using somewhat of a different instrument is called random positioning machine, but the overall effect is the same. To reduce gravity and see what is the effect of microgravity in these biological specimens. So the way we have structured our pipeline here goes like this. We have first the RAMP system, Rapid Aging Microgravity Protocol. Then we do signature validation. What does that mean? Well, it means that we have a bunch of transcriptomics data and proteomics data in our simulated microgravity that we want to confirm it also happens in humans in space. So we gather all the data from crew members that have gone in space, and we compare those and basically run an overlapping analysis. But we also incorporate a third aspect of this, which is aging cohorts on the ground. We want to be able to mimic what happens in aging on the ground. So having these three buckets and the intersection between those, we pull all the biomarkers and we infer mechanisms, and we also develop our interventions to see if we can reverse those things. And so when we pull the compounds, that the gene compound enrichment analysis, then we screen them in the RAMP system. So we go back to this system to see if they work, right? And the last thing we do, so this is kind of backwards, is disease mapping. So very different from what traditional drug discovery does, which is I have a disease and I need I have an indication, I need a drug. In this case, I have a very interesting system to understand accelerated aging in a dish, and then I do disease mapping to see which diseases are going to be affected by my system. And so it's ideal not just to us for developing our own drugs, but also for the pharma industry. So the pharma industry has about 10,000 compounds sitting there with no indication. So they do have compounds with no particular indication, so we can help them repurpose those. That's one of the models that we're working on, licensing the technology to pharma. So the promise really is that as compared to traditional drug discovery, we are able to accelerate the discovery of these drugs and by using human, so no animals in this case, and by about 15-fold in the first, say, 3 to 4 years of target validation, compound screening, lead optimization, and all that stuff. So by about 15 to 20-fold. So I'm going to show you some of the results that we've gathered over the past year and a half in the company as part of the Cosmica effort. In immune organoids, cardiac organoids, as I mentioned, also neural organoids and PBMCs. And these are cells obtained from the blood bank. Okay, so first off, the immune organoids in the left-hand side. What you see here is the responses to the flu vaccine of an adapted immune organoid that can resist microgravity simulation as a function of age. So it thus recapitulates what happens in aging, the fact that individuals of older ages do not respond as well to the flu vaccine. And so we're using these to understand the process and target these things. And we have some compounds, as I'm going to show you, that are doing quite well at reversing immunological aging. Now, the next one is cardiac organoids, and in this case, these are produced by iPSCs. They come from induced pluripotent stem cells, and we use from healthy patients. But we also compared with dilated cardiomyopathy, for example, or other conditions associated with heart aging and heart failure. And I'm going to show you just one simple example of our monitoring of our little 1 millimeter hearts. It looks really interesting. What we're seeing now is that when you take cardiomyocytes alone, the effects of microgravity are null. If you take cardiomyocytes and endothelial cells and you build this more coherent organoid, then you have a huge effect of microgravity. So we know already now that the effect of aging, and I can show you all that data, comes from the endothelial cells communicating with the cardiomyocytes. And there's somewhat of a bradycardia effects with cardiomyopathy signatures. I'll just stop there. The other types of organoids are also a little bit larger. These are 2 millimeter brain organoids that we study by using this Axiom machine. And you can see how they have this firing potential that we can study before and after microgravity simulation. And there's also very interesting findings there. We, as I mentioned before, the clock of brain aging applied to Alzheimer's disease and other dementias is overlapping to a large extent with what we see under microgravity. More data. I hope this is not very overwhelming. I'm going to go relatively quickly here, but with 25 hours of simulated microgravity, we see acceleration of aging in different aspects on biology of aging. So the first one is the inflammatory age I mentioned before. Microgravity or zero gravity compared to 1G, and you have an increase in inflammatory age by about 40%, only in 24 hours of microgravity simulation. We have a reactivation of different bacterial infection here. We also have reactivation of viruses under microgravity. We have increase in cellular senescence. You probably have heard that some cells become senescent with age, and they accumulate in tissues, and they produce a large number of inflammatory biomarkers. And so we have increased cellular senescence by about 35%. And then we also have the epigenetic clock measured in these samples. And what you can see under microgravity, there is also an increase in epigenetic age as early as 24 hours or 25 hours microgravity simulation. Overall, the immune response, when you stimulate those cells after microgravity, they don't respond. So they don't produce gamma interferon, they don't produce alpha interferon. A very interesting finding, many people ask me, so what's the mechanism? Why all these things happen in conjunction? What we believe it happens very early on in the response is a remodeling of the actin cytoskeleton in the cell. And that drives, to a large extent, that mitochondria falls apart. And then you have obviously the consequences of dysfunctional mitochondria with production of reactive oxygen species, inflammaging, and all those downstream effects. So we are at least recapitulating 7 of the 14 hallmarks of aging. And it's not that we're not mimicking the other ones, it's that we haven't measured those yet. So I think we're in a good shape to say that we are now using this model to search for anti-aging compounds. So let me walk you through the GCA package, which is the gene compound enrichment analysis, a little bit better. So we have drug bank with maybe 100,000 compounds, Links 1000, this is a NIH consortium, FoodDB, so we also use food ingredients and compounds. And we take all these and run the interactome analysis that I mentioned at the beginning of this talk. And so with that interactome analysis, we have lead compounds that can reverse those signatures. This is just one example of a COVID project I did in the past to repurpose compounds for COVID-related alterations in the immune system. And what you see in columns will be, I can barely see here, columns is the genes, and in rows are the different compounds that were pulled by our algorithm. So that's to identify hits. And then what we do next is to test those in the microgravity simulation. So we go from hit to lead compound, and we have been very lucky because our top compound shows that 70% of the differentially expressed genes under microgravity are reversed by using this compound. And what are the pathways that are most affected? Very interesting pathways, and as I said before, the last step in this process is the disease mapping, and that's what we do here. So we have seen that we can probably mimic or recapitulate a signature of hepatic fibrosis, osteoarthritis, cardiac hypertrophy, and we can reverse those pathways with the compound of interest. So the pipeline looks like this. It's really an evolving target as we are discovering more diseases that are mapping what we see in the different organoids. We just ordered a mutated line of organoids that resemble what happens in Alzheimer's disease. And we have now identified 10 different compounds that we're testing. We have 23 different compounds for immunological aging. That's underway, the testing of these things. And as I said before, in 18 to 24 months, we're aiming to submit this as an IND to FDA. OK, so I think there's going to be a lot of room for questions, Jennifer, if it's OK. So I'd like to thank part of my team here that I have at the Buck Institute. As I said before, I'm also a 1000 Immunomes Project director here at Stanford. We secured another 18 million to continue the 1000 Immunomes for the next 5 years. But again, if we don't bring private sector in the middle, it's very, very challenging to translate things into the bedside. So we do other things other than science. As you can see, my team goes for surfing and partying, and now we're planning a retreat in Tahoe. It's going to be really exciting times for us. And then we also grow watermelons. So if you have the opportunity to be at the Buck Institute, you're going to see our watermelon growing. I think this is very anti-aging. Not the watermelon itself, the fact that you're growing it. Right? It's very important. And then the last thing is IMPay. Thanks to this amazing architect who helped design the Buck Institute. It's a tremendous venue. It's really fantastic. If you have the opportunity to go to Marine, I invite you to come to our institute. He designed the Louvre pyramids in Paris, the Islamic Museum in Qatar, JF Kennedy Library in Boston, and many other hallmark buildings. So with that, I will probably stop here and take questions. Thank you so much for your attention. Thank you. Yeah.
OK. 2 questions. Number one, do you get specimens of blood or anything else from people who have strong family histories of longevity on one hand, or certain diseases like Alzheimer's and heart disease on the other? And number 2, I've read that the spike protein from COVID virus, which is used for both the vaccine and which causes, the most serious consequences of the infection, is largely its effects are mainly by causing inflammation. And is anyone studying the long-term effects as far as aging and inflammation in people who've either had the vaccine or the virus or both? Thank you.
Yeah, wonderful questions. I would ask maybe if you can introduce yourself before asking the question. That will be helpful.
I'm sorry. Roz Shorenstein, retired internist, and I also have a PhD in biochemistry.
Thank you so much. I do have answers for both questions. The first one related to extreme longevity. the data I showed you on the collaboration with the University of Bologna and centenarians only considered 19 centenarians. We're now working with hundreds of centenarians in collaboration with Nir Barzilai at Einstein to analyze their inflammatory levels and many other aspects associated with their genetics in these Ashkenazi Jews that have extreme or exceptional longevity. We even put together a program grant where we're using advanced artificial intelligence to get to a mechanistic view. So you have the genes, you have a phenotype, which is longevity, and you have other phenotypes, as in multiomics in the middle. You can build what's called Mendelian randomization and have an idea of how mechanistically related, so correlation, causation, we go for causation, right? Those genes are. So we are looking at centenarians. They exhibit a really interesting microbiome, for example. I can just say it now. The CD4 T cells are behave completely different than the general population. So there are some peculiar aspects of being a centenarian. And as I showed you before, they exhibit a lower inflammaging overall. On the other extreme, the disease individuals, we are working actively with Stanford collaborators as well as with UCSF. So we have a program in Alzheimer's disease on the ground, not just the space stuff, in brain aging mostly, in collaboration with Joel Kramer at UCSF. So he has collected thousands of samples, and these are samples PBMC, PAXGene2 for RNA sequencing, serum, and you name it, with a large cohort of thousands of individuals that were followed over the years. So this is a longitudinal study of aging. And we have one paper that was just published in the PNAS and another paper that looks at the trajectories. So we're taking those extremes of trajectories. So one thing is to start, quote unquote, biological older, and a very different thing is to evolve and how those rates change over time. So that's the second paper that we're cooking. And then the second question, can anyone remind me of the second question? I kind of elaborated on the other stuff. I'm a little bit sleepy.
Of COVID,
yeah, COVID, yeah.
Yeah,
so
causing inflammation.
Yeah, sure. Thank you. So the other program, one of the programs we have is in infectious diseases, particularly interesting in COVID. We have a collaboration with Kari Nadeau here at Stanford and Mark Davis as well. We collected samples from COVID patients at discharge, but also 6 months out. And we were able to analyze proteomics, in particular this biological aging clock of the proteins that we gathered from another public domain database, which is a paper that was published by Tony Whisker here at Stanford, actually. So we took all those measurements, about 1,500 proteins. We derive a proteomics clock, and it was not surprising to see that COVID patients, severe patients, show an accelerated proteomic clock that lasts for at least 6 months out after a discharge. So I guess one thing that I should mention is that long consequences on proteome aging in COVID patients is a reality, is expected. We've seen it. And so, yes, there are long-term consequences of COVID. Yeah. Yeah, it's moderating here.
Yes, in your, I am wondering, in your cohort of 1,000 people that you're getting together to do this testing of inflammation, do you, out of that 1,000 people, you're going to have a number of different factors that are causing inflammation. Have you developed an algorithm that you can, if one factor comes up, you do this. If another factor comes up, you do that. The thing that impressed me on your slide was the number of lymphokines that you have there. And do you have adequate blockers for those in the study that you do, or is that part of the algorithm? That's my first. My second question is on the organoids. More and more, they're using organoids, and I've always wondered how much of that data you get from organoids is transferable to the actual human condition, and I'd like for you to comment on that.
Yeah, yeah, wonderful questions both. Let's see. Let me ask the organoid question first. There is some good mapping with diseases that we see in patients. The organoid systems in general are, the technology is really in its infancy. We are using them as if they were great models for what happens in a human. The big problem is that they're not connected with other organoids. So you study an organoid in isolation, but things don't work in isolation in humans or any biological system. So we now have a grant that we submitted, that we're going to submit, sorry, this is in the process of being written, to understand organ-organ communication. And we thought about using organoids, but it's fairly impossible. So there's this concept of multimorbidity, one disease causing other diseases and propagation of that signal. You cannot possibly study that in organoids. So for some cases, they are very good surrogates of what happens in vivo. For other cases, not that much. So the immune organoids are really good at recapitulating an immune response. And they come from tonsils, from older patients, most likely from sleep apnea, and younger patients due to recurrent infections. I hope that answers the question. And then the second question, still so sleepy, was about? Yeah, so the factors, were you referring to biological factors or lifestyle factors? Because it was not clear to me. Is it the biological factors that we're measuring? Like the chemokines? Okay, so, because I didn't, with the 1,000 immunomes, we cannot address, to the extent that I wish, the lifestyle factors, but I have a whole program on lifestyle, and I can tell you more about that if you're interested later on. But the biological factors, so we did see a bunch of adipokines and chemokines popping up as being significant for the inflammatory age, which is, I don't know if it's expected or not, but they're not the suspected ones. It's not IL-6, it's not TNF-alpha. They're not part of the equation. They're measured, but they don't contribute. Whether or not we have blockers of this, we're looking at medication history. And that's probably the best way to do that. We don't have an interventional study for the 1,000 immunomes where we can, say, give an antibody against CXCL9 and see how things change. I wish we could. That's all part of the company. And again, it took us 15 years to develop this diagnostic test. Now we're bringing, the Bayers of the world and the Paladin Capital, and then in two years we run 1,000 people interventional study, and we're almost ready to go for an IND. So it's important to bring that part into the equation. But we're looking actively at medications and interactions between medications. And it's really interesting because it seems like statins are going into the opposite direction. I think probably most of you know some studies that have shown anti-inflammatory role of statins. Well, we don't see that. We see quite the opposite. But I should stop there. Yeah.
All right. Yes.
Unfortunately, we only have time for one more question.
Oh, no.
Stay on schedule.
All right. Thank you so much. My name is Ravi Chandra. I'm a psychiatrist in San Francisco. Thank you for this beautiful biological deep dive. I have a transdisciplinary question about psychosocial impact, and I don't know if you know the answers to these, but what are the effects of adverse childhood experiences and zip code on cellular markers of aging? In other words, are there some people on Earth who are in fact living in space?
Well put. So I don't have data to show you, but we, as part of this inflammaging effort, I have created the National Inflammaging Institute with Mike Schneider here at Stanford, with Ruslan Metsitov at Yale, Luigi Ferrucci at the NIA. It's a large effort that seeks to find cures and mechanisms of inflammaging by funding particular projects on that area. So as part of that effort, we are developing now a platform to understand that question. So in other words, what are the social and the exposome, the term, I'm not sure if you're familiar with the term exposome, but the exposome is the collection of biological, physical, and chemical exposures we have throughout life, and that's part of our lifestyle. How those contribute to inflammaging or inflammation. And then, so that is going to be an app. And the one thing I want to mention is that when you build this app, you can keep people engaged. And the idea is to really promote health, not just for a few, not just for those who can pay, but for billions. And so using this app, I'm going to be able to tell you where your inflammaging may likely come from. Is it stress? Is it sleep? Is it a pollution? I'm going to get your zip code for sure. So we can estimate pollution, and we can estimate your scoring system for inflammaging just by looking at this survey. Now, how do you track those changes? If you're promoting a healthy living and particular lifestyle recommendations personalized for that individual, how do you track changes? I mentioned very briefly the idea of a selfie. So we now have a mean, we are probably 1 year out to finalize that project, to estimate your biological age, not just for the immune system, but other organs, by looking at a photo of you. Okay, so how do we easily monitor the health of the individuals across the board by looking at a healthy selfie, I call it. So we would be able to integrate that into this platform, and hopefully we can improve the health of the entire population without the constraints that that we have in general as a society. Yeah.
Thank you so much.
Wonderful. Thanks.
Thank you.
Thank you, Paul.